Hello, erudites, and welcome back to Next Level Catch-ups 2.0 for General Biology. In the last lesson, we discussed about the cell cycle and mitosis and how it functions and its main purpose. In today's video, we'll be discussing about meiosis, another type of cell division, but this only happens in what we call as gametes. So, meiosis is a kind of cell division that only happens in gametes. It comes from the Greek word meion, which means to make small. The meiosis cycle um, makes the original number of chromosomes to only half. So if let's say we have 46 chromosomes in humans, after meiosis, the number of chromosomes will only be 20, 23. So it aims to separate not just sister chromatids but also homologous chromosomes, meaning to say that it the traits are going to be separated. So in meiosis, it's also divided into two processes, meiosis one and meiosis two. So in meiosis, as we mentioned earlier, the daughter cells or the cells after the process happens has only half of the original number of chromosomes. Now, this only occurs in these gametes, such as the egg cells and the sperm cells. In the sperm cells, it's the spermatogenesis, and in ovaries, it's called as the oogenesis. So, why do we need meiosis? So, in the fertilization of a zygote, then the DNA material of a zygote comes from both cells, that being the haploid sperm and the haploid egg, wherein we take half of our parents' DNA, half of both our parents' DNA, so that we could produce a new, a new, a new, what do you call this? A new organism. So a haploid versus a diploid. So a haploid it refers to set of chromosomes. So it's only defined as the letter N. So this only occurs in gametes, sperm, and ova. While a diploid, on the other hand, refers to a pair of chromosomes, one from each parent. It's depicted as the 2N and only happens in somatic cells or body cells. For sister chromatids, or homologs, as I explained earlier, sister chromatids share the same genes and alleles, meaning to say that it's the same genes or same traits with the same manifestations. Homologs, on the other hand, are the same genes or the same traits, but with different alleles or manifestations. So let's get into the overview of the stages of meiosis. So in meiosis, it's similar to it's similar in mitosis, where it still follows the interphase and the PMAT cycle. So after this, it just repeats into meiosis too. But that's we'll discuss that later. So as mentioned earlier, in meiosis, it still follows the same PMAT phase. So in the first meiosis one, the homologs separate. So in the prophase one, unlike in mitosis, it has five stages or substages. The first one is leptotene. The leptotene is when the chromosomes condense over, and then the zygotene, on the other hand, is when the chromosomes produce tetrads or a pair of two chromosomes, and then they prepare to cross over in packetine. In packetine, this is when the genetic material crosses over between non-sister chromatids, so that it would cross over the different alleles. And then after that, we have the diplotin or the synapses, which is the um, actual trade-off of the genetic material. And then in the diakinesis, this is when the chromosomes have already finished uh, crossing over, distributing the different traits, and the nuclear membrane disintegrates. That's basically it for uh, the first prophase, so prophase one. So as mentioned earlier, that's pretty much it for prophase one. We'll proceed with metaphase two, metaphase one rather. So in metaphase one, it's pretty simple. The tetrans line up in the middle, and then the spindle fibers uh, begin to attach to the tetrans in preparation for the next phase, which is the anaphase one. After metaphase, anaphase one comes to split the tetrans to move the chromosomes to the opposite poles, and by the end of this, we'll remain with uh, pairs of chromosomes, not four pairs of chromosomes, but rather two pairs of the chromosomes. So in this one, we we can see that the chromosomes are still in its chromosomes form, in comparison with mitosis, where by this phase it would already have been sister chromatids. In this, After this, we'll come to telophase and cytokinesis. In the telophase, uh, the chromosomes decondense and the nuclear envelope may start to form. So during this as well, we can observe that the cleavage furrow is already beginning to form in preparation for the next stage with it, which is the meiosis two. Here, the daughter cells are haploid cells. So the telophase one still contains the chromosomes in its chromosomes form, but they begin to decondense. So after that, we'll move on to meiosis two, where the sister chromatids separate. So in this stage, we can see that once once the chromosomes, the tetrans have already separated in meiosis one. In meiosis two, we'll begin to separate the sister chromatids so that it could produce half of the number of uh, so that it could produce cells that contain half of the number of the original uh, cell count in the parent cells. So that's primarily it for part one of our lesson regarding meiosis. I'll be handing you over to Kim to discuss meiosis two and the different stages of meiosis two. So I'll be handing you over right now.
What is up, erudites? It's me, Kim. Thank you, Kaz, for explaining the first half of the lesson. Now, we'll be tackling the second phase of meiosis. First, we have the prophase 2. The prophase 2 is the first stage of the second meiotic division. At this point, two haploid cells exist, each with half the original number of chromosomes. But each chromosome is still made of two sister chromatids. During this stage, the chromosomes condense and the nuclear envelope breaks down in both cells. A new spindle apparatus begins to form in each cell, oriented perpendicularly to the original spindle in meiosis 1. So the three key points to take note in this particular phase is that the nuclear envelope disintegrates, the chromosomes condense again, and the centrosomes move to opposite poles. In metaphase 2, the chromosomes in both cells align individually along the metaphase plate or the equatorial plane of the cell. This alignment is random, allowing for further genetic variation. The spindle fibers, which originated from the centrioles, attach to the centromeres of each sister chromatid, preparing them for separation. So the main thing to keep note in this particular phase is that the spindle fibers align the chromosomes at the metaphase plate or at the center. In anaphase 2, it is the stage where the sister chromatids finally separate. The spindle fibers shorten, pulling the sister chromatids apart and moving them toward opposite poles of the cell. Once separated, each chromatid is now considered an individual chromosome. This separation ensures that the new cell will receive a complete set of chromosomes. So the main thing to take note of this phase is that the chromosomes, sister chromatids, they separate due to the spindle fibers and then they move to the opposite poles. Lastly, in telophase 2 and the cytokinesis, these are the final stages of meiosis. In telophase 2, the chromosomes arrive at opposite poles. They condense, and new nuclear envelopes form around each set of chromosomes. Concurrently, cytokinesis divides the cytoplasm of both cells. This division results in four distinct haploid daughter cells, each with a single set of chromosomes. These cells are genetically unique from the parent cell and from each other. So the main thing to take note of this is that the chromosomes decondense, the nuclear envelope forms again, and then while this happens, the cell divides into two, and the results are four haploid daughter cells. Next, we have the results of meiosis. We have four. Number one, gametes form or the sex cells. Male gametes are called sperms. Female gametes are called eggs. Next, four haploid cells. Haploid meaning only 23 chromosomes. Four haploid cells with one copy of each chromosome. It also results into one allele of each gene and various combination of alleles for different genes. Next, we'll be discussing the three main ways genetic variation is created. First, we have the crossing over. Crossing over is the exchange of genetic material that occurs between homologous chromosomes during prophase 1 of meiosis. As chromosomes from the mother and father pair up, they can swap segments of DNA. This process creates new, unique chromosomes that contain a mixture of genetic information from both parents, ensuring that the gametes produced are not simply a copy of the parental chromosomes. Next, number two is the independent assortment. The independent assortment occurs during metaphase 1 of meiosis, when homologous chromosomes pairs align up randomly along the cell center. The orientation of each chromosome pair is independent of the others. This random arrangement allows for an incredible number of possible combinations of maternal and paternal chromosomes in the resulting gametes, making each one genetically distinct. Third, is the fertilization. Fertilization is the final step that maximizes genetic diversity. It is the random union of a sperm and an egg cell. Because each of these gametes is already genetically unique due to crossing over and independent assortment, the combination of any two of them creates a new individual with a one of a kind genetic makeup. Lastly, for our generalization, sexually reproducing organisms have a second form of cell division that produces reproductive cells with half the number of chromosomes. This process is called meiosis, and without it, humans, trees, beetles, and all other sexually reproducing organisms would be vastly different than they are today. And that ends the last lesson of general biology this midterm. Why is this important, you may ask? Well, meiosis is crucial because it's the process of cell division that makes sexual reproduction possible, and it is the primary source of genetic diversity. Without it, life as we know it, with all its variety, wouldn't exist. That's all. Thank you for listening, and study well, erudites.